G'day, welcome to Nevadia. Boo-ba-doo-boo-ba-doo-boo-loo-loo-loo-loo-loo. How you going, everyone? The other week, in typical 2020 fashion, this happened. This is scary. The hyperbole, that is. For those people who are driving right now, I am talking about the outbreak of the bubonic plague in Mongolia that was reported on by the international media last week. I realise that I've only done one microbe video, which you will find up here. Maybe. Considering microbes are some of the coolest things on this planet, I thought I probably should do another one. By the way, this is going to be packed with a lot of technical info, so I've linked in the description below the script so you can reference it if you need it. Also, due to the amount of conflicting information I have found, I encourage you to take some time to do some of this research for yourself. This week, I am diving into Yersinia pestis. It is the microbe that causes the bubonic plague. I'll also explain what an outbreak of this microbe means, and I will attack some of the geopolitical issues that have come up. Because this year, outbreaks haven't been used as a justification for discrimination against an entire population at all this year. Firstly, let's discuss the outbreak itself. It started in Inner Mongolia, a weird this is Mongolia, but it's not actually Mongolia, but it is in fact China, but the more ethnic Mongols live in this than Inner Mongolia, but it's not Mongolia because it's stuff and geopolitics. Anyway, if you want some more information, I have linked a video in the description below. If you want to understand it more, then I can actually explain what it is. Since Inner Mongolia is officially part of China, this outbreak is officially Chinese. I'm going to refer to it as a Yersinia pestis infection because the term bubonic, or black plague, has way too much emotional baggage, and I want to divorce the easily treated disease we experience now from the disease that decimated entire continents back hundreds of years ago. Back then, we didn't actually have the medicinal knowledge that we do now. So what is Yersinia pestis? It is gram-negative, non-motile, rod-shaped bacterium. I won't go into what gram-negative bacteria means, but links to it are below. Non-motile means that it won't move around by itself, and rod-shaped means it's, well, rod-shaped. It evolved from another disease-causing bacterium, Yersinia pseudotuberculosis. The difference between the two is that Y. pseudotuberculosis is spread through water and soil, whereas Y. pestis is transmitted from animal to animal and, scarily, through aerosols as well. That is why it was so quick to spread around the world centuries ago. Scientists think that the microbe originated in the Gobi Desert, and some evidence suggests that it started in the Neolithic decline approximately 6,000 years ago. DNA evidence published in 2015 suggests that it infected humans in Bronze Age Eurasia, and there were some genetic changes in the microbe which increased its virulence about 4,000 years ago. It was found to be the causal agent of the bubonic plague by Alexander Yersin in 1894, which is where the genus got its name. If you can't remember what a genus is, it is a category of biological classification which ranks between the family and species. For example, humans are of the genus Homo and our species is Sapien and our subspecies is Sapiens. Neanderthals are of the genus Homo like us, but their species is Neanderthalensis. Going back to the genus Yersinia. There are several species and subspecies, 3 of which are pathogenic or disease-causing types that I will talk about later, but the most famous one would be Yersinia pestis medievalis, which caused the Black Plague. There have been 3 historically significant waves of this disease. According to some sources, the first pandemic started in 540 CE, lasted for 60 years and killed up to an estimated 40 million people. However, other sources say that the first pandemic spanned 210 years. I don't know why there's this discrepancy. I suppose there's just some differences in methodology. The second pandemic started during the 1300s and is thought to have killed between 50 to 200 million people. In Europe alone during this outbreak, 25 million people died, which was a third of the population at that time. The last outbreak started in 1896, killed approximately 10 million people, and officially ended in 1959. However, some could argue that we are still in the third pandemic, as ongoing outbreaks of Yersinia pestis have been detected worldwide almost yearly. One reason for the worldwide spread is that the last outbreak started in Hong Kong and was carried to other continents via shipping routes. Untreated, a Y. pestis infection can be deadly 30 to 100% of the time. Treated infections can still cause death approximately 8 to 15% of the time. Okay, now that we have established that Y. pestis is deadly, how does that kill you? Most people know that Y. pestis is transmitted via a flea bite. The microbes then travel to the lymphatic system via macrophages, an immune cell that numbs up foreign bodies. Yersinia pestis then multiplies in the lymph nodes, which is where the bulbs from the armpits and the groins come from. By the way, those bulbs are a symptom of the plague, hence bubonic plague. Sometimes these bulbs rupture from the pressure caused by the multiplying microbes and they get released into the bloodstream. When that happens, it progresses to septicemic plague, which is much more deadly. Septicemia is an infection of the bloodstream, so yeah, it's pretty nasty. Even with our current medical technology, it is still a very serious disease. After its progression to septicemia, it may travel to the lungs where it then becomes pneumonic plague. If the disease gets to that, you pretty much have a 0% chance of surviving. Yay! If you want to learn more about the epidemiology of Y. pestis, I've linked that and many other sources in the description below. Remember when I said there were 3 different types of Y. pestis? The first one is called Antigua, which caused the plague in the 6th century. The second one, Medievalis, caused the Black Death in the 14th century, and Orientalis, which was responsible for the current outbreaks. What makes it so deadly? Well, allow me to talk some sciences with you and hopefully you can keep up. This is high-level stuff, so I'll do the best I can, and if you can't follow, that's totally fine, you are definitely not stupid. A reminder that I've linked my script in the description below if you want to read along with me, or you can use it to Google something for clarification. In pathogenic or disease-causing microbes, there is something called the virulence factor. It is not only a part of bacteria, but it is also present in viruses, protozoa, and fungi. Virulence factors are anything that helps the pathogen cause disease and are what we use to measure its virulence or likelihood of causing a disease. Virulence factors also allow the pathogen to avoid being destroyed by the immune system as well. These can be molecules, toxins, surface coats, etc. Virulence genes are the bits of DNA or RNA that code for virulence factors, if that makes sense. Yersinia pestis has multiple virulence factors, and this is where I'm going to go up a level on my sciences, so bear with me. The first type of virulence factor it produces are 2 types of toxins, an endotoxin and a murine toxin. An endotoxin is a lipopolysaccharide. Scary word, so I'll break it down for you. A lipid is a type of fat. A saccharide is a type of sugar. Poly means many. So a lipopolysaccharide is basically a chain of sugars attached to a fat molecule. Endotoxins are found in the cell wall of disease-causing microbes, usually gram-negative ones like our friend Y. pestis. When the cell dies, it breaks up or lyses, releasing the endotoxin. Have you ever heard of someone getting sicker when they started antibiotics before they got better? That's because the microbes making them sick had an endotoxin. As the antibiotics destroyed the microbes infecting their bodies, the concentration of the toxin they release on death increases. After a few days, the patient will start to get better, unless the toxin load is too high and they die. Unfortunately, this is not unheard of in severe infections of endotoxin-producing bacteria. From what I could see, a murine toxin seems to be only found in Y. pestis, but I only did a short search, so it is possible it could be found in other microbes too. The word murine relates to anything to do with the genus Muridae, such as rats, mice, and other rodents. When injected, they produce local swelling and necrosis, or death of the surrounding tissue, paired with systemic effects on the peripheral vascular system and the liver. The peripheral vascular system is the blood vessels that exist outside of the heart, by the way. It also attacks the immune system by infecting macrophages, or the immune cells that eat the pathogen. Usually, once the macrophage eats the pathogen, it digests it. In the case of Yersinia pestis, the immune cell eats the microbe, but instead of destroying it, the microbe instead infects the cell, similar to the human immunodeficiency virus, or HIV. This means the immune system is weakened by the infection. For those who are interested, this is done by the F1, V, and W antigens. Basically, the F1 antigen prevents the bacteria from being eaten, and the V and W help it infect the immune cells. Type 3 secretion systems inhibit the production of cytokines, which are proteins that are used in cell signaling and are especially used in the immune system. Basically, cytokines are proteins that tell your immune system to do its thing. If the production of cytokines are inhibited, your immune system's response is reduced. In the case of Yersinia pestis, the inhibitions of cytokines is shown by a reduction of inflammation, which is a usual response to infection. Another virulence factor Y. pestis has is the reduction of opsonization, or the process of marking the microbe as a pathogen and therefore telling the immune system to make antibodies to fight said microbe. It also causes the blood to not clot properly, which spreads the disease further in the body by degrading fibrin clots. Fibrin is the protein that causes the blood to clot. Despite this, it also contains coagulase, which causes fibrins to collect around the microbe, helping to protect itself from the immune system. It's pretty much the perfect disease. It spreads quickly, easily, and has a very high death rate, even with treatment. If you've kept up with me here, then congratulations. Here is a photo of a cat to help your brain catch up. Get it? Catch up? Please make sure you subscribe to my channel to make up for all the people who just unsubscribed to me for that joke. But why am I saying it's the perfect disease, but not to worry about it? Well, it has a cure. It's called antibiotics. They're cheap, easy to manufacture, and easy to access. Another reason why we really don't need to worry about it is that we understand a lot about this disease. In places where the disease is endemic, doctors would know what the symptoms look like and are able to issue alerts quickly. We know where it comes from, which is the hunting and eating of marmots and other rodents or mammals. Simply knowing where disease vectors are, preventing the spread of a disease is made much easier. People at high risk of infection are using antibiotics as a prophylactic or a medicine used primarily for disease prevention. The now famous hydroxychloroquine is known as a prophylactic because it is a drug taken to prevent malaria while traveling. There's also a vaccine. While it's not very effective against the pneumonic form and there are significant side effects which has led the US to stop the use of this vaccine, there is still a vaccine in the case of a worldwide outbreak or biological terrorism. One of the earliest examples of biological warfare using the plague was by Tatar sources. They were laying siege to 14th century Kafar and they catapulted their plague victims into the city. This of course started the pandemic, which ended up killing 50 to 200 million people. In more recent times, Japanese forces dropped plague-infested fleas over Chinese cities in World War II. In 1995, Larry Wayne Harris and William Leavitt tried to release Yersinia pestis into New York. This microbe has many animal reservoirs worldwide, so it isn't very difficult to obtain if needed. It is also extremely virulent, has a low infectious dose, has a high transmission, and is easily genetically engineered to resist antibiotics. Because of these factors, it is a disease considered to be a concern for biological terrorism, and multiple vaccines are being developed. Besides, why pestis outbreaks happen naturally every year somewhere on the planet, so it's worth developing a vaccine for it anyway. The disease is spread through aerosols, just like the current plague we are going through. However, the biggest danger is when the disease has reached the pneumonia stage. By that time, the person will have already shown symptoms of an infection. Before the pneumonic stage, it is unlikely to spread through aerosols. But, just like all aerosol-borne disease, a mask will greatly reduce the chance of a spread. So wear a mask! Without current medical treatment and antibiotics, Y. pestis is a horrible infection. However, with early intervention, it is in fact a very manageable disease, which is why we don't see worldwide pandemics like we have with our current one. Now that I've explained what Y. pestis is, how it works, and why it is not actually that scary, I want to talk about the media coverage of recent outbreaks. Let's not pretend these headlines aren't capitalizing off the fact that this outbreak originated in China. This leads into the narrative that all global outbreaks begin there. They don't. Clearly. For example, there was a global outbreak of the Zika virus that originated from the island of Yap in 2007. Yap is found in the Western Pacific Ocean. The disease itself was first identified in Uganda in 1947. You also have MERS, which was a global pandemic that started in April 2012 in Jordan. It, too, is a coronavirus and is mainly contained within the Middle East. However, this disease can spread in the same way COVID-19 does, but with less death. It also can't spread from asymptomatic people like COVID can, as far as we know, which is really why you need to wear a mask even if you don't have symptoms. One of the reasons why China is the originator of a lot of global pandemics is its size. It is approximately 1/50th of the world's landmass and is the 4th largest country in the world. It has rainforests, deserts, cities, you name it. Just by simple chance, it will produce a larger portion of the world's pandemics. It is also home to 20% of the world's population, and a lot of them are densely packed into urban centres. This allows disease to spread much more quickly than you would see in less dense populations, such as rural areas. There are some factors in China that would cause novel diseases to develop due to human activity, but in the case of Yersinia pestis, this outbreak is purely natural. So this hysteria about another pandemic coming from China is just a little bit racist. Second of all, as I've mentioned several times before, Yersinia pestis is found in most areas of the world, meaning this outbreak could have started pretty much anywhere. In fact, there have been reports of a squirrel testing positive to Yersinia pestis in Jefferson County, Colorado on Saturday the 11th of July 2020, which, scarily, is the first time it's been detected in that particular county. It's currently not endemic in Australia, by the way, but it is a reportable disease if found. Oh, quick definition of endemic. It is a disease that is self-sustaining within a geographical area. For example, the zoonotic Hendra virus is found in bats in Australia and is endemic within the bat population. It passes from bats to horses to humans and can be deadly. The Hendra virus isn't endemic in horses and humans because the disease can't self-sustain. This is due to vaccinations and quarantining measures, which breaks the chain of infection. Since it's difficult to immunise the bat population, the disease is continuously being spread amongst the species. Going back to Yersinia pestis, there are outbreaks yearly at some point on the planet. The majority of the outbreaks are due to interacting with infected animals, coming in contact with contaminated tissue, and eating infected animals such as marmots. In the US, outbreaks are linked to squirrels. In fact, there was a small outbreak of the plague in the US in 2017, where 11 cases were recorded and 3 people died. This links back to my earlier comment about the use of a disease to discriminate against people of a certain ethnicity. The headlines about the outbreak in Inner Mongolia links to the very fact it's from China. They play to the fear around COVID-19 to clickbait the hell out of it, thus making the newspapers or media outlets more money. There's no two ways about it, and it's despicable. Our current pandemic originated in China. Its origins are linked to human activity. It is around because species that normally would never have interacted with each other were in the same vicinity. The virus was able to combine and share genetic information with other viruses in the same family. The difference between the current outbreak of COVID-19 and the outbreak of Yersinia pestis is that Y. pestis is not caused by crowding animals that shouldn't be together, but was caused by hunting animals who were infected. It is no different to how some people would get salmonella poisoning by eating raw or undercooked chicken or eggs. The only difference with Y. pestis and salmonella is that salmonella is not contagious. Overall, we shouldn't be afraid of this new outbreak. They happen frequently all over the world. It is easily treated. It doesn't spread very far. We know how to cure it. Please don't buy into the hype over the new disease that will kill us coming from China, because it's not going to happen. And this hysteria is out there to scare more people and get more clicks. Okay, now that I'm sure you're slightly less terrified of this newish, oldish plague exiting China, let me ramp up that horror for you. There are strains of Y. pestis showing signs of antibiotic resistance. Yes, I just said that. In 1995, there were 2 cases of people in Madagascar contracting a strain of Y. pestis that had high-level resistance to prophylactic antibiotics. Luckily, these 2 people, aged 16 and 14, were able to recover with oral and intramuscular injections of antibiotics. You know the needle they jab in your ass if they can't get rid of an infection? Yeah, they underwent that daily for 4 days. Antibiotic resistance is a known side effect of overprescribing them. If you have a virus that is not accompanied by a bacterial infection, do not take antibiotics. Unless, of course, your doctor tells you to because of risk of infections or other information that I know nothing about because I'm just a YouTuber and I am not a doctor. Please don't sue me. All I'm saying is listen to your doctor and please don't ask them for something you don't know you need. Other diseases that are developing resistance to antibiotics are Mycobacterium tuberculosis, which of course causes tuberculosis, and Staphylococcus aureus, the microbe known as golden staph. You also might know it as MRSA or methicillin-resistant Staphylococcus aureus and can cause septicemia and other life-threatening diseases. So the fact that Yersinia pestis is developing resistance to antibiotics is not actually all that surprising, which is why a vaccine is extremely important. Another thing to be scared about is the window for Y. pestis infection is widening due to climate change. Areas of China where it's endemic are also getting wetter. This allows more vegetation to grow, which in turn gives rodents more food. More food for rodents means more rodents. More rodents means more fleas. More fleas means more Yersinia pestis. The drier parts of China are also closer to Europe. Currently, there are no reservoirs of Y. pestis in Europe, according to the highly respected journal Proceedings of the National Academy of Science, or PNAS. Yes, I know I'm not very mature at all. Therefore, Europe is at risk of reintroduction of this disease, thanks to climate change. Conversely, in the current wetter parts of China, more rain, weirdly, leads to less plague. They haven't quite figured out why this is the case, but changes in the climates may cause shifting in the geography of the frequency of the outbreaks of this disease. The same thing is happening in parts of America, not just the aforementioned Jefferson County. In 1999, yes, that long ago, a paper was written talking about an increase of precipitation will also increase the frequency of plague outbreaks in southwestern USA. Also, if you want more diseases to be worried about being spread due to climate change, tuberculosis, anthrax, and smallpox are also on that list. Yes, smallpox. You know, that disease that was declared eradicated on the 8th of May in 1980? Yeah, that one. It could come back due to climate change. Can we just stop destroying the planet? Like, please? But a bit of good news to round off this depressing video. You might have heard that Yersinia pestis has caused a bit of a headache for creationists. This is because it is thought to have caused selective pressure on the frequency of a mutation in European populations. This is known as the CCR5 delta 32 gene mutation. What it does is it removes a protein that is used in the immune system as a receptor for chemokines, which includes cytokines. Quick reminder, chemokines are a family of proteins that are used in cell communication. These proteins work a little bit like a lock and a key. If a cell receives a signaling protein, like a hormone, it attaches to the cell via the receptor protein. If it's a fit, the cell lets the signaling protein inside. The CCR5 delta 32 gene mutation removes one of these receptor proteins. If you are homozygous for this mutation, meaning that you have this mutation on both copies of your alleles, you don't have this receptor. This receptor is found on a lot of your immune cells, including your macrophages, or the cells that nom things up, and your T cells or the angry killing cells. The Black Plague enters your immune system and proliferates through the macrophages. HIV enters the immune system through T cells. It has been found that people with the heterozygous CCR5 delta 32 mutation do not have this particular protein covering their immune cells, or less of them. Heterozygous means that the mutation is only on one allele, but not the other. I've linked a video below if you're confused, including about what an explanation of what an allele is, which is a totally different topic. People who have the CCR5 delta 32 mutation on both alleles display a significant decrease of viral load and a slower progression of the disease HIV. Since it is possible that Yersinia pestis used the same pathway to enter the immune system, it may have caused some natural selection that favoured this mutation, as people who would have had some immunity to this disease survived the pandemics. This is suggested by a high incidence of the mutation in European populations in areas that were hardest hit by the Black Plague. But there is a growing number of scientists that suggest that it could have been another disease that causes selective pressure, due to some areas having a high incidence of this mutation in surviving descendants that weren't hit hard by the Black Death, and other areas that were hit hard but they don't have this mutation. Other diseases to have caused this are suggested to be smallpox or an Ebola-like hemorrhagic fever. Again, sorry about this very information-heavy video. You might want to watch this a few times. I have, of course, linked all of my information below, as well as the script of this video for the people who need to read this while watching it. Microbes are fascinating. They are the key to our salvation and to our downfall. It's up to us to decide which one. And don't forget, scandium, iodine, nitrogen, and cerium. I just want to say a very special thank you to my wonderful patrons, especially my $10 Redback Spider patrons, Aided Furball, Lauren Hart, Mary Civitano, Amanda Vogue, Ross Devereux, and Atheist Pastor. I also want to say a very special thank you to my $20 Platypus patron, Paul Butler. I hope I have made you significantly less terrified of this disease, because, as I said, it's not as bad as what we normally associate it with. It's curable, doesn't spread very far, and is easily treated. For now.